about the basic unit of life, the cell. Most people start there. I think it's a good place to start. When we take a look at cells, though, they're very diverse. That is, we have different types of cells. In this case, we have a sperm cell contrasted to, let's just say, a neuron. And you can, I think, agree with me, they're very, very different in structure. I've got a couple of others thrown in there. A red blood cell, which, if you look at it on edge, has a biconcave disc, so that's a very different physically, if you will, structurally. Epithelial cells, they don't always have cilia on the top of them, but this is an example of an epithelial cell, let's say probably from the small intestine. And then we have a smooth muscle cell. There are three types of muscle. This is just one of them. There's also skeletal and cardiac muscle, but they're just illustrating one type of muscle cell. So I think you'd agree that they're very diverse in terms of their structure. Also, I think you would agree that they're very diverse in terms of their function. Because what's the function, for example, of a red blood cell? Okay, most people say carry oxygen. It's a good response. Turns out they do a bit more than that. They even carry a little carbon dioxide for us, but it's very good. That first response is what most people say, and that's typically what I was looking for. And how about a neuron? What does this do?
Transmit signals.
Okay, signals, or we can say carries nerve impulses. And so they're very, very different, I think you'd agree, from carrying oxygen to carrying a nerve impulse. I don't know, I didn't add them up. They say we have about 75 to 100 trillion cells. Quite an enormous number, and about 200 different types of cells. The good news is we're not going to be trying to find all 200, but we will get to quite a few different cell types. I don't really have a number for you, but it's not going to be anywhere near the 200. So finishing off, again, we know that cells vary in shape and they vary in function. I thought what we'd do is take a look at a typical cell. This cell, what I've done is thrown everything in it. And I'm going to name the parts. And then what we're going to do is take a slow look at each of those parts. Not in great deal of detail, but just have a general idea of what's going on inside the cell. Again, I want to emphasize, based on what we already saw, that you would agree that this isn't like some of those cells that we just looked at before. So we're just going to take this particular diagram and think of it as a typical cell, knowing that cells can be very different in terms of physical structure and function. Let's get to naming some of the structures. Inside we have the nucleus. We can see ahead of time that the nucleus actually has a double membrane. Also, we can see the nucleus has pores, physical pores through which materials can enter and exit. There's also a structure, which we'll talk more about, inside of the nucleus called the nucleolus. Associated typically with the nucleus, continuous with the nucleus, and even with the cell membrane is the endoplasmic reticulum. You'll notice this part of the endoplasmic reticulum has little dots on it. Those dots represent ribosomes. We can probably jump ahead and say that ribosomes are the sites of protein synthesis, and so we're going to come back to this. Also notice that there's a possibility of having smooth ER inside of a cell. Smooth ER is, well, kind of simple in a sense. It's in comparison because what? It does not have the ribosome attached to it. So we can then say there are really two types of ER. Rough, that would be endoplasmic reticulum with ribosomes attached, and then we have the smooth where we don't see any ribosomes attached. And again, we're going to come back to these things later. Nearby, near the endoplasmic reticulum, typically associated with it, we see the Golgi apparatus or Golgi body. Also notice in this diagram they do it very nicely. You can see that from the Golgi apparatus we see these little vesicles, if you will. We call them endocytotic vesicles, or actually I should say exocytotic vesicles. And we're going to move materials out of the cell, secrete them in other words, through a process called exocytosis, and we'll encounter that also later. We see the mitochondria. Notice that the mitochondria, that's plural. If I just point to one, that's a mitochondrion. Notice it has a double membrane, an outer one and an inner one. We also can see centrioles. Actually, one centrosome consists of two centrioles. We have lysosomes, peroxisomes, as soon as I find them, peroxisomes. And this wouldn't be true for every cell, but we see microvilli on this particular cell, and a variety of tubules inside of the cell, microtubules and filaments. Again, all of these things we're going to look at in a little bit. Let's start somewhere, and the first place to start, I think, would be the cell membrane. Now, this is not a picture that you have. Remember I said I was going to provide pictures sometimes that are just there so that you don't get swamped with pictures in your notes. This represents actually two cell membranes, here, and you'll notice in both cases that they look like they are composed of, or at least you can see like a double line. This would be the intercellular space, interstitial or intercellular space, and then we would have inside of each cell. So this would be called intracellular, extracellular. But you could technically call this extracellular because we are between cells. We would call it intercellular rather than extracellular. But it is outside of the cell technically. This electron micrograph, colorized just for us, of course, but this electron micrograph shows us a magnification that's probably a couple 100,000 times. That's quite a bit. And that's about all you can see. So in other words, we've never been able to physically look inside of a cell membrane. And yet we know a heck of a lot about it. More and more, I would say, in the last 20 years than all the years before that. But nevertheless, that's all we can see through a microscope. And this, of course, like I said, is an electron microscope. And they can magnify some of them well up or close to 1 million times. And even if we use one of those, we're not going to see much more than those two lines. So a question I might have for you is, how on earth are we going to study the cell membrane? I'll just throw it out at you. How are we going to study it if we can't see it?
Do stuff to it.
What do you mean?
You shrink with like alcohol, make it expand with water. It has to be hypotonic water. It is the correct
Okay, so
term.
what you're saying, break it apart?
Not necessarily, but I mean, you know, you have to mess with it, run
I'm
experiments
going
on
to
it.
stop and ask you a question, because obviously not me, I know some of the things that they do, but we can take some red blood cells, I can get them very easily, and we can take all the hemoglobin out, and then we can get a pound if we want of membrane, and we can then analyze it. And that sounds good, but what happens when we start to analyze something? How do we know if we take all of the membrane of a red blood cell, how do we know that when we analyze it, we're going to get the right answers, you might say? What's going to happen when we start to analyze it?
You have to first make sure it's pure, you have to run tests on whatever you're
Okay.
testing.
I guess what I'm getting at is when you start to take something apart, don't you start taking some things off that shouldn't come off the membrane, etc.? So they've had a really rough time to study the membrane over the years, and I can assure you that that's probably, I don't know about 100 years, but you probably have close to 100 years worth of research, and it's still an active area, so you could go to graduate school and you too could get a PhD, if you will, in some aspect of membrane transport or receptor information, because really the information that we still need is endless. We've got a lot of information, but on the other hand, there's an awful lot to go. So it's very difficult to take the membrane apart. And so we do analyze it, and we have to learn or get techniques that allow us to take apart the cell membrane without ripping it all apart. In other words, what if I take this protein and I cut part of it off, so to speak? Then I haven't properly analyzed the membrane. And there's a lot of ways to take this into account, and the bottom line is they are able to get us kind of a pretty decent representation. Actually, this goes way back, which is a long time ago, right to 1972. Nicholson and Singer came up with what they called the fluid mosaic model of the cell membrane. It was kind of revolutionary in its time, if you will, and now, of course, it's not a theory or anything. Everybody accepts it. And in this diagram, you can kind of see that idea. You can see a membrane, may not understand its fluidity yet. We'll talk about that. And you can see these purple proteins, if you will, interspersed within the membrane. And it's a mosaic, a pattern, in other words. It's constantly changing because it's fluid. And how do we get that fluidity? Well, you can see that the membrane is composed of phospholipids. This little gray circle and the two little tails on it represents a phospholipid. So think of the head, the little ball that's been drawn here, as being phosphate, and then the tails as being lipid or fat. Now, phosphate is PO4. That's how we could write phosphate down, right? Phosphorus and 4 oxygen, PO4. And water, of course, is H2O. My point being that when something has oxygen in it, probably a good chance it might be soluble in water. So phosphate is soluble in water. And you can see what has happened. We're 2/3 water. All of us, men and women, are approximately 2/3 water. So what has happened then, the phospholipid has oriented itself in a certain manner. I think you know at home, oil and vinegar don't mix, right? We shake them up, and if we wait a little while, they all settle out, they don't mix. Well, that's the same thing here. And so what happens then is the phosphate head, which is soluble in water, orients itself towards the water in the extracellular space, and the other phosphate head from the other phospholipid orients itself towards the intracellular space, which is also about 2/3 water, if not more. Okay? So we wind up with what's called a bilayer of phospholipid. Interspersed in that phospholipid, we have a variety of things. Yes, we have cholesterol. And you'll notice in the notes I mentioned that cholesterol accounts for about 20% by weight, and I think 50% of volume. That's a lot, isn't it? So did this artist draw this properly? Do you think it was proportionate? No, we probably should have even more cholesterol in this picture than we have. So I know we always think of cholesterol as kind of evil, if you will, but you can see it's part of our cell membranes. All of our cell membranes have cholesterol as a building block. Now, I'm not advocating you go out and have a lot of cholesterol, because obviously there are issues with cholesterol, but the point is it is a natural material in our body. It is used, it's part of us, if you will. It's just that it's true when we have levels in the blood that are far too high, other things can happen. We can get fatty deposits and other things can go from there. So we definitely don't want to be taking in too much cholesterol, but we shouldn't say, hey, I need zero cholesterol, but my body, as it turns out, can synthesize cholesterol, and does synthesize it.
So would you classify cholesterol as HDL or LDL or a mixture of both?
No, I wouldn't. HDL refers to high-density lipoproteins. In other words, that's a protein that's carrying fat. Low-density lipoprotein refers to, again, another special protein that's carrying fats in a different direction. So these two chemicals carry fats in the blood, and we think of the HDLs as the good guys,
right,
and the LDLs as the bad guys. But this isn't really this is not a protein. This is pure fat, cholesterol. Okay, good question, though. So we also have proteins. Notice that the proteins that stick into the cell membrane, they don't have to even travel all the way across, but any protein that sticks into the cell membrane, these are called integral proteins. They didn't show us one that partially sticks in. Perhaps this one right here, it's hard to really tell. But again, the protein does not have to extend all the way across the membrane. If it extends all the way across the membrane, and it's true, all of these do, then we can call them a transmembrane protein. Because what does that mean, right? Trans, it tells us that we're going across the membrane. We also see some peripheral proteins, which are typically attached to other proteins that are already within the cell membrane. So are peripheral proteins within the cell membrane? No. By the way, there are thousands of different proteins probably in a cell membrane. And when you look at this picture, I'm glad we have a picture like this, but my point is that it's really an oversimplification. And I just think you should realize that it's not just, oh, look at this, 1, 2, 3, 5 or 6 proteins. No, there are literally thousands, and they're so different. They're not all the same. The cell membrane is a very complex structure. I don't think we could solve every problem with our bodies, but if we knew the complete structure of the cell membrane, there's no doubt that we'd have the cures for a heck of a lot of cancers and other things. I don't know about everything, but it would be pretty close, because that cell membrane regulates things in and out. Right? If you can block out, we'll call it the bad guys, so to speak, a virus or something, then you are going to have a lot less problems. So we're always learning more and more about the cell membrane. Now, what they didn't show us in this picture, I'd like to at least mention it, because when you get to physiology, we'll talk about this, depending on the teacher. I know you will talk about it. You may talk about it a lot or a little, as long as you know about it. I would say in the last 20 years, one of the most important things, like discoveries, or one of the areas where we've had the most advancement is in our knowledge and discovery, and then our ability to discern the structure of receptors, receptor proteins. When hormones interact and other chemicals interact with the cell, they typically interact with a receptor, and that's typically a protein. And it can be very complex, and it's amazing now because they can actually, using X-ray diffraction and other techniques, they can actually figure out the structure. But you might say, well, that sounds good. No, you don't do it in a weekend. It may take somebody a degree, a PhD or something else, to characterize the structure of that protein and how it works. So if we have thousands, it really takes an awful lot of work to do this. We take it for granted, but it's coming along. And the more knowledge we have of receptors, the more knowledge we have, or more ability we'll have to actually, you might say, cure disease or prevent it. And some of you may have heard this when you take medication for, let's say, high blood pressure. You might have heard the term beta blockers, calcium channel blockers. Maybe you haven't, that's all right, but you may hear this terminology. So here's a channel protein. That's what they're trying to illustrate here with the passageway. We don't know what it is, but we could say that this is a channel for calcium, let's say. And so we can introduce a medication that's a calcium channel blocker that somehow can interact with that channel and stop movement. So there's a lot going on. We're just looking at this in a descriptive way because in physiology you'll get to look at some of these other things in more detail. Yeah.
So does our body make all the cholesterol that cells need, or do we need to ingest more to reduce?
We can make it. We could make it all. So you could really reduce your cholesterol to 0 and you'd be fine. It's a little hard to do, but I guess if you were a vegetarian, you would be having extremely. And if you don't eat any poultry or eggs, you could really reduce your cholesterol to pretty close to 0. But yeah, we can synthesize it. The liver will synthesize it with synthetic pathways there. In fact, what they do with some of the medicines, like Lipitor and some of those medicines, what they do is they block a key enzyme in the liver for the conversion into cholesterol. So what you're really doing is blocking the production of cholesterol, and therefore it drops, and your blood level drops.
So would that be the same for like diabetics, sugar, or if they have to,
What do you mean?
like does the liver, whatever the insulin, if that's just injecting sugar into the blood?
Well, insulin is a special hormone that helps the glucose to get into our cells. And of course, that's not a fat. That's would be a carbohydrate. Okay, let's move on a little bit more. Notice that attached to some of the proteins we have a carbohydrate. Now, these little squares that you see here are literally six-sided, which tells me they're really glucose. So this is a string of glucose molecules, and it's attached to a protein, so we call it a glycoprotein. And then if we have these glucose molecules attached to a fat, as soon as I can find them over here, the phosphate, which of course is part of the fat, we then have what's called a glycolipid. These are very interesting, and I've got a picture of them. It turns out that those glycolipids and glycoproteins form, you might call it a sugary coating on the surface of the cells, and this is called a glycocalyx. So as a result of all of that, those glycolipids and glycoproteins, we have literally sort of a sugar coating around many cells, and it's believed to be involved in cellular recognition. So we don't know enough about that glycocalyx now, but one of the thoughts is that it's involved in cellular recognition. We also can see some other things. For example, we notice that we have some filaments of the cytoskeleton. You might say, what keeps the cell together? Why doesn't it just collapse? And actually, it's like a building in a sense. It's got a scaffolding, an internal scaffolding of filaments and tubules that's just indicating some of those filaments that are attached to the inner side. And by the way, there is no doubt that the outside of the cell, this will be extracellular, and this intracellular. There's no doubt that the structure of the outside is different from the structure of the inside of the membrane. So you can't say reverse, and they're really quite a bit different as a matter of fact. Are there any questions on that? All right, let's go past the glycocalyx. And I put this up here because it's a different author's view of the same thing. And again, you don't have this picture, remember I said I'm going to give you lots of extras, because it's repetitious. And so what I'd like to do with you is simply let you look at this picture, and then we'll play around with it a little bit, because something's going to happen. We're going to look at proteins in a moment. But you can see the glycoprotein. Notice that they've drawn the integral proteins a little bit differently. And this is very interesting, because this represents one of those transmembrane proteins that's involved as a receptor. And you can see that it actually travels through the membrane actually 7 times. And so that's a very special protein. So this artist, I think, did a good job. There's our cholesterol. What do you think about the amount of cholesterol? What about this artist?
More.
So we still need more. It's just, again, it's just an illustration. Now let's take a look at the peripheral proteins, or rather the integral proteins first. Hopefully you picked them all out. Next, let's take a look at the peripheral proteins. Take a look and see where you think they might be, and let's see if that works. All right, so those are the integral and peripheral proteins, and I just wanted to show you something. Your own textbook will have a different picture. And you might say, why are all the pictures different? And I think you should, simple answer, we don't want to be caught plagiarizing, right, when you write a book. So you have a different artist, and the artist looks at the other person's picture, and they all look and kind of try to make something a little different. I'd like you to see more than one picture, so that's why I wanted to give you this extra picture. It's kind of a nice one, it's just a different approach to the same thing. There are different types of proteins. We have channel proteins, transporter proteins, receptor proteins, cell proteins that identify us, there's markers, adhesion proteins. And I thought what we would do is spend most of our time, since we want to be more descriptive, is to stay and take a look at junctional proteins. So over on, I think it was about page 3, it talks about the cell membrane proteins, and I mentioned some of them. We're going to focus on junctional proteins because, again, anatomy is supposed to be a little bit more descriptive. The physiology of the others would come next semester, if you will. So let's take a look at some of these proteins. This diagram represents the junctional proteins, all of the junctional proteins that we're going to talk about. We have tight junctions, we have desmosomes, and we have gap junctions. So this is sort of a review or an overview, and now we're going to take a look at the specific structures. Let's take a look at the tight junctions first. While we're here, let's identify a couple of vocabulary words. What's a lumen?
Space.
I'll buy that if you finish it. You're going to add more to it.
Like a hole within a vein, like the hollow part of
Okay.
the vein.
So it's a passageway within a vessel, or it's, if you want to look at it in a very crude term, it's the hole in the hose, right? And it's a term that you will get to use. We could talk about the lumen of a catheter, right? And we might even ask you, is it open or closed? And the word for open is patent. So we might say, is the passageway patent? Is it open and clear? Or is it blocked? So simply you would say, someone would ask you, is your syringe or the cannula patent, meaning open? And you would go yes or no, depending on what you have found. So this is the lumen. Let's take a look at think about the digestive system for a minute. There's a stomach and a small intestine. Let's just say we're right there in the small intestine. The lumen of the small intestine. Everyone knows where we are then? Right? Okay, good. Because that word's important because but many times we're going to hear that word and we need to know what it means. I think you might agree that if we're in the lumen of the small intestine, it's a pretty hostile place, isn't it? Is it sterile? You don't eat sterile foods, do you? No, clean food, I hope, but it's not sterile. It's not sterile. So this food comes in through your stomach. It's where the digestive process starts in the stomach. We continue on in the small intestine. We've got various digestive enzymes that are going to help break down all the food molecules, the fats, carbohydrates, and proteins. And what else might be in this mix? Was there any bacteria on the surface of that food you ate?
Yes.
Yes. It really was, unfortunately, you might say. Is there bacteria on your arms right now, even though maybe you scrubbed yourself, had 3 showers this morning already? Yes, still bacteria on there. So there's no doubt that there's a mix in here of enzymes, chemicals, and bacteria. And the simple question, would you want this to get into the bloodstream? No, not in that form. So we have tight junctions between the cells, which is going to limit what materials can get through, if any, through those tight junctions. Bacteria, no. Many of the harmful chemicals, no. How are we going to get the good stuff in? Well, the epithelial cells, these special structures, these would be called microvilli. These microvilli and the cells themselves will literally, through a variety of active and passive processes, pump or move the goodies, if you will, into the cell and then, of course, into your blood vessels. So we'll exclude the harmful materials because of these tight junctions. Just because I've shown some cells here with tight junctions, you might say, well, could they have any of the other junctions? And the answer is yes, but what predominates? Tight junctions. Tight junctions in this particular example. Let's take a look at another structure called the desmosome. The desmosome, I like to think of it like the body's Velcro. Take a look at the way it's designed. These filaments interlock, and it's a sure close thing to Velcro. If you've ever seen Velcro up close, it's got all these little hooks on the end, and all you have to do is just barely touch those hooks, and they kind of grab on each other. It's not permanent, but it's pretty strong, and that's exactly what we have here, quite a strong connection. So where there's going to be a lot of stretching and movement, we would want to have a lot of desmosomes. So the skin, for example, the cells that form the skin would have lots of desmosomes that hold things together. Also, not shown here in this diagram, but we also are going to need a type of desmosome to hold your cells to the what's called the basement membrane. Notice what's going to even hold our cells onto something. We have something to hold them between each other, desmosomes, and there's even a special type of desmosome that holds the cells to a basement membrane. You have to ask yourself, why don't my cells just, pull apart? Because I don't always think about, well, how does this work? Well, because we have such structures to hold everything together. So this doesn't mean that there are absolutely no other types of connections here with junctional proteins, but again, predominantly in the cells of the skin, we would expect to have desmosomes. Another junctional protein is called a gap junction, and the actual junction, or you might call it a little connector, so to speak, that is part of the gap junction, they call it connexon. And look at the diameter of the passageway through the gap junction, 1.5 nanometers. What's that in angstrom units? Can you figure that out? Should be able to work a little bit with those things, those micrometers and nanometers and angstrom units. You really should be able to work with those, all of those, a little bit more easily than I think you might be doing it right now. So how many angstroms is that? Well, it's 15. There's a 1 decimal place between a nanometer and an angstrom, right? You remember an angstrom is a 10th of a nanometer. So try to practice a little bit of those conversions. It's just helpful because then you have an idea also of what the size of something is, what it really is. So this is small, very small. So what can pass through? No large molecules. You're not going to get glucose or any large molecules like that passing through. But there's ions and very, very small molecules that can get through. So a sodium ion, for example, or a potassium ion would be able to move through a structure like that. Do they help hold the cells together? Yes. But they do have a special function. So you wouldn't really have these in the skin, for example, but I'll tell you a place where we have a pile of them, and that happens to be between cardiac cells. Our cardiac cells, heart cells, are held together by the connexon, and the reason for that is we have to be able to have one cardiac cell communicate with the other. In other words, a nerve impulse literally is going to be able to pass right through the cardiac cells, not instantaneously, but from our point of view, with the way we look at it, it is just about instantaneously, but it's a wave of contraction. We're going to be able to coordinate that contraction because we have a way to get these ions, which actually allow us to move the electrical impulse from one cell to the other. There, they actually form a physical structure called an intercalated disc. And we'll take a look at that a little bit later on in the semester. So are there any questions on those? Now in your notes on the next page, there is a list that says transport of materials through the cell membrane. I have no intention of going into detail on this because this is really physiology, but I thought I would mention the fact that when we move things through the cell membrane, there are a variety of ways to do this. And we have what are called passive processes, which do not require something called ATP, and we have active processes that do require ATP. That's a word many of you I know already, adenosine triphosphate. You heard it in high school or somewhere. You heard it maybe in terms of muscle contraction, exercise, but you'll hear the word, again, even today you're going to hear the word a couple more times. So you don't have to memorize any structures of it, but it is, you might call it the energy currency of the cell. It's the way the cell can do things. It has to have ATP. If you were to poison ATP, and you can do this, there are chemicals that the an individual can use in a laboratory and actually poison a cell's ability, in a laboratory dish, of course, not in us. So we can actually do this, and so that's why we know some processes are active and some are passive. Because if I put a certain chemical in, the chemical happens to be called ouabain. It's ouabain. If we use ouabain in our dish or in our experiment, that blocks all active transport. And so if something still happens, then we know it must be passive, because this would block all active transport. The active transport, there is what we call simply active transport. There's vesicular transport, that also involves ATP. There's endocytosis, whereby we actually engulf material to bring it into the cell. Also used to be called pinocytosis, that uses ATP. Exocytosis, which you saw earlier, and I'll show you again. How do we secrete materials from the cell? That requires ATP, so the process of exocytosis, getting materials out of the cell, requires energy. Phagocytosis is something actually it comes from the Greek and it means cell eating. And phagocytosis is a bigger event where we might ingest bacteria. So we have cells that can move through the skin called macrophages, and they can attack any bacteria that might have been pushed into the skin from, let's say, a splinter or something else. Phagocytosis happens on a much bigger level. In other words, it's a larger event, if you will, because a cell literally moves around another structure and actually engulfs it. So that's called phagocytosis. So all of those are active. So if I put ouabain in the mix, what happens? Stops it. Nothing happens. On the other hand, diffusion, where things move from high concentration to low. Filtration, which is just a separation of materials by particle size. Osmosis is the net diffusion of water. All of these things, and facilitated diffusion, are all passive. You put ouabain in, and what happens? They keep functioning. And notice the word in that last one, facilitated diffusion. This involves a transmembrane protein, and we don't quite understand this completely, but what's very interesting about it is that a chemical will literally fit into a certain spot within this transmembrane protein, and then it will help it enter the cell, so it facilitates. And it's kind of hard to explain something that can facilitate without using energy, but it does. So that one hasn't really been, no one really, that I know, has fully nailed down 100% how facilitated diffusion really works. Why? Because we can't see these membranes change their structure when we take a look at them. Because when you start looking at cells in electron microscopes, you've already preserved or, you've already altered the cell, and so you really can't be sure of what's happening very often when you look at something under the microscope. What you would like to do, which seems impossible right now, I said seems, I'm careful. I didn't say it is impossible. What seems impossible is what you'd love to do is see that protein changing in front of your eyes, right? You'd like to see the chemicals somehow interact, and it's moving, and I would say, I'll just leave it. Right now it seems impossible, but who knows what the next technology will bring. And, we might be able to actually see something like that. Let's move on and take a look at some of the structures within the cell in more detail. So you looked at that first picture where we looked at all the different structures. Now we're going to focus on a couple of structures. Your own book, again, you notice I didn't necessarily take these pictures from your book, but every book's got pictures that should be somewhat similar. The nucleus has been called the control center of the cell. Now, I say it like that because I guess it's one of those things, I don't know who said it, but someone said you put little quotes around it, I guess. In every book, everybody says it's the control center of the cell. And so we have to find out why is that. Well, inside the nucleus houses your DNA, your chromosomes, that which you inherited from your parents. And some people, I think of DNA as something that, hey, I use that when I need to have a kid, right? And we're going to have a baby or something. Okay, now it's time to use my DNA. But actually, you're using the DNA every second of the day. You wouldn't live, believe me, for whatever at all if your DNA could not function in every single cell, brain cell, and everywhere else. So within the body, all your body cells, in other words, all cells except for sperm and eggs, so that's an awful lot of cells, right? All cells except for those that have 46 chromosomes or 23 pairs of chromosomes that you've inherited from your parents. And in all those cells, that genetic information is directing the second by second or microsecond by microsecond activity of that cell. And I say, how could that be? Well, the DNA, which I'm just pointing, we don't have it drawn there, but the DNA within the nucleus has a code, right? We call it the genetic code. I think it was in 2003, I think, thereabouts, where we actually were able to put together it was called the Human Genome Project, and they were actually able to figure out the sequence of base pairs in DNA. They even did it ahead of time. By the way, when I know you've probably watched CSI or one of those shows. There's so many different CSIs, right? And they analyze a person's DNA, and by the night shift, they've already got everything set. Before the show's over, they've got everyone's DNA analyzed, and you know, in the real world, sometimes they even have to send it out to other labs. It's just not something that you can just do in 10 minutes. Maybe it will be, but right now it really isn't. It's a lot faster than it used to be. There's no doubt about it. But I want to emphasize that they are not really monitoring all of your genetic information. Because look, it took all those years, right, till 2003 just to do it once. And they were doing DNA testing before that, weren't they? When was the first DNA testing that showed up in, I guess you might say, where everyone become aware of it? Does anyone know? It wasn't 2003.
Wasn't it World War II?
Well, what I'm really thinking of is the O.J. Simpson trial. It was the first time we really saw DNA out in not the discovery of it. Discovery was back in 1953, but I'm talking about the fact that where did we all get to see it or hear about it? Maybe your parents, depends on your age. That was what year was that? 98. You know, so most people, you may have been young kids, whatever, but a lot of you probably, saw a part of that or heard about it. That's for sure. And my point being what? That was the first time really where they had to teach the people of the jury what DNA was and everything else. And in other words, nobody really was, the scientists understood it, but they were trying to convince the jury that yes, we can use DNA. And at any rate, my point being that they did. That's what did we say? 1990.
96.
94. Okay, whatever. 94. Any of those. But if it's 1994, we didn't even do the human genome, right, until 2003. So in other words, what they do is they chop up big pieces. And the way, even though they're not even chopping up every little base pair, they're able to analyze big chunks, and it turns out that we're so different that even with the big chunks of analysis, we can be 100% sure that it's someone's DNA and not somebody else's. So that DNA then has a code, if you will, and that code directs the production of RNA, which is, a lot of RNA is stored in the nucleolus, and then the RNA can move out of those nuclear pores and go somewhere out of the cell, which we'll look at in another picture or two, and then we can make a protein, and proteins regulate the cell's function, because most enzymes are proteins. Enzymes regulate chemical reactions. So bottom line, your DNA is running the show. No, it's not thinking for you. It's just running the metabolism of the show, all the chemistry and chemicals that have to, ultimately, they're not doing it directly, but indirectly because of the production of RNA. The DNA stays in here, good reason, right? That's the good stuff. We want to keep it safe, and RNA can move out, and we can keep making more RNA as we need it. There is a very definite nuclear pore, very clear and observable nuclear pore. It's also been called the central library of genetic information. Sounds good? All of those are those catchphrases, and they're out there, and there's no reason why we can't use them also. Let's take a look at a structure that's associated typically with the nucleus, at least the nuclear envelope. It's continuous with the nucleus itself and also with the cell membrane. See it? I show it all the way from the nucleus all the way out to the cell membrane. This is the endoplasmic reticulum. It's a membranous network And of course, if it has little, we'll call them dots, or in other words, if it has ribosomes on its surface, then it's called rough ER. And if you don't have ribosomes, you have smooth ER. Just as a little note, the most extensive smooth ER in the body is in skeletal muscle. We'll look at that another day. All of that tubular system of muscle, and it's extensive. So there we call it the sarcoplasmic reticulum, but it's all smooth, does not have ribosomes on its surface. And by the way, ribosomes can be attached, as you see here, or they can be floating freely, and we'll talk about them in a minute. The ribosomes are the sites of protein synthesis. So think about this, if we make a protein, it can move into these membranous structures, the ER, and then move to another location in the cell before export, or for that matter, we can even keep something in the cell. Notice, if you take a look at your notes, the ER has quite an extensive number of functions. It can modify, it can detoxify certain drugs and chemicals. It stores materials for export, so it has quite a number of functions. Let's next take a look at the association of the ER with another structure, because it's associated with that structure. As a matter of fact, it forms that structure, and that's the Golgi apparatus. Proper name for the Golgi apparatus, because obviously an individual is named after an individual, as you can see, Camilio Golgi, and I guess we haven't taken that name away yet. I say that because your textbook especially, and it's a good textbook, the McKinley book, but those authors have a habit of really taking away all the older terminology, and I have a little bit against that. I think you could put a parenthesis in there, because there are still many doctors and young doctors out there who would really be using both terms, if you will, for some of these things. And I don't know if every doctor is being trained only from your book, so to speak, without the old. And so I'll talk about more of that as we go along. So your book is great, nice. I'm just saying that they have a habit of getting rid of the old, which I know we have to do at some point, and then only using the new terminology. So I'll assume that they, if you have your book out there, they still use the word Golgi apparatus here. Correct me if I'm wrong on that. Now the Golgi apparatus can be described as a series of flattened vesicles. In other words, it's like taking a ball and flattening it out, so this is what we mean by a flattened vesicle. Notice that it's formed from pinching off from the ER, so its source is the ER. And then the material, that's the protein typically, that's been moved into the ER, moves through those passageways, makes its way into the Golgi apparatus where it is stored and modified so that we can chemically modify the material and we store it. Let's say, insulin as an example. Insulin is a protein hormone. We're going to have the messenger RNA. In other words, we're going to have a lot of protein synthesis, and then that insulin is going to move into the Golgi apparatus where it will be modified, and then finally it will be secreted by the use of a secretory vesicle and exocytosis and move out. Since we need insulin all day long, there's going to be a continual production and regulation of the production of insulin. If you were to look inside of a cell, a pancreatic cell, it would have an absolutely extensive rough ER. And that tells you that this cell is probably making something for export, and sure enough it is insulin. So a pancreatic cell, when you take an electron micrograph, it's got dots all over the place, rough ER all over. It looks like the whole cell is almost made up of only rough ER. Any questions on that? Let's take a look, perhaps out of sequence, but let's take a look at mitochondria since it comes next, and then we'll go back to our ribosomes, I think, in another picture or two. The mitochondria have one of those terms that we can kind of put in quotes that someone said sometime, a long time ago. They're called powerhouses of the cell. And that's because most of the cell's ATP is made in the mitochondria. The mitochondria, again, that's plural, or here are they mitochondrion. But in mitochondria, there are double membranes. So here's the outer membrane, And then we have an inner membrane that folds. Notice how it makes folds. Within these folds, then we have shells. And the term for shells is cristae. Crista would be one, and plural would be cristae. So we've got shells or cristae within the mitochondria. Within these shells are what are called oxidative enzymes, which allow us to basically produce ATP. These oxidative enzymes work on certain molecules that enter the mitochondria, and then the end result is they produce ATP. So mitochondria are very important to us because they make a lot of energy. If you were comparing, let's say, a muscle cell to a skin cell, which one might have more mitochondria? Yeah, the muscle would have more mitochondria, very extensive mitochondria, because obviously with all the work, every time we move just a little bit, we're going to be using ATP. And yes, we can make ATP outside of the mitochondria too, but the truth is we make most with the mitochondria, so they're very important to us. A couple of interesting things here. Mitochondria self-replicate. They have their own DNA. Yeah. Well, it looks like, according to what we see here, we have about 37 genes. They're not ours. Well, they are, but they're not. Obviously, it's in us, so of course you can say, if it's inside my cell, they're mine. But they're not in the nucleus. That's where the genetic information is that you've inherited from your parents, is within that nucleus. And so the mitochondria literally mitochondria have their own DNA that allows them to replicate, produce more mitochondria. Now, there's no doubt that there is a link between our DNA and the mitochondria. In other words, the mitochondria isn't all by itself deciding when to divide and multiply. There are obviously going to be triggering events, chemicals that interact with the mitochondria that trigger the process. But once the process is triggered, the mitochondria they're going to use the DNA that's present within the mitochondria. But there definitely has to be a relationship, a control.
So our DNA from the nucleus tell the mitochondria how it's
Yes, yes, but not in a very roundabout way. It wouldn't be. Our RNA has to be made, and other things have to happen. Yes, but you're right. This just can't happen because if a cell's going to divide, which is also a very regulated process, we have to have more mitochondria. We can't take the original number of mitochondria, split them, and every time keep splitting them. So we have to replicate them. Obviously, that's under control. When the World Trade Center went down, you know they had to analyze DNA to find out, literally, so that people could have closure. Yes, my whatever husband, wife, whatever daughter, whatever it was, yes, was in that Trade Center. And people just, whatever, it's like psychological thing, whatever it is, we want to have the closure, right? We want to know, yes, so-and-so was there, you did find the remains, that type of thing. And they still haven't found all of the remains. I don't even know, I think they're still working on it too. And they use all types. They use DNA, of course, and they also even use mitochondrial DNA also. Anything they can to help them identify that individual. Now, notice what this says. Mitochondrial DNA is what? Only inherited from your mother. So what happens is when the sperm literally attacks the egg to try to penetrate through the outer barriers, let's just draw this. There's the mitochondria. There's the DNA. Once the head of that sperm penetrates the membrane of the egg, all of the sperm are locked out, and these mitochondria, or the middle piece of the sperm, is isolated. That is, it is not allowed to enter. So the only DNA, mitochondrial DNA, that's available is what? Maternal. Doesn't matter whether you're male or female, your mitochondrial DNA is always maternal. Because these mitochondria never get there. Okay? Kind of interesting. Any questions on it? Let's take a look at a because they're linked. Do I do a different sequence? What did you want, what was I going to do next? Maybe lysosomes? Okay, let's see if we can go find lysosomes first. There we go. And go back, try to take it stay in sequence. I noticed that out of sequence here, so I don't even know where I am anymore.
Lysosomes are alive.
Okay. Mine are, I don't know where they are. That doesn't matter. Let's take a look also at lysosomes. This is another structure within the cell. They're very small structures. They were discovered in 1953. As you can see, they're about 0.2 to 1.2 microns, which is quite small. They contain hydrolytic enzymes, and those I mentioned about 40. It doesn't matter what, it's a lot. We have a lot of what are called hydrolytic enzymes. Notice the word hydro and lytic. Lytic, lysis, breaking. Water breaking. So in other words, many bonds are produced by removing water when we synthesize things. So what we're going to do here is add water and break things apart. You have at least over 40 different hydrolytic enzymes within the lysosomes. They were discovered back in 1953, which is a long time ago. And you might even ask, why didn't they find them before then? What happened back then? I know it's a long time ago. I know, it's history. I know. I realize that. But, yeah, there's certain, but when the electron microscope came out, now it was out, I'm sure, in the 40s. I know it was out in the 40s, but after a while, some biologists said, hey, there's a new instrument out there that can really magnify. If we can only use it for biological samples, wow, it would be great. And so they figured out a way to use it for biological samples, and then we were able to start looking at things with an electron microscope, which has a totally different magnification than our light microscopes in the laboratory. I guess today's our first day with the microscope, so today we'll talk about resolution and things with the light microscope in lab class, and I'll bring up electron microscopes again. But electron microscopes can magnify several 100,000 times. It's unbelievable. And that's with clear detail. That's with separation of materials based on particle size. In fact, we use the term resolution, which is the ability to separate fine detail. And so those electron microscopes can do that. Most hospitals will have a scanning or electron microscope at the hospital. There are certain diseases that we can actually determine by using the electron microscope. So it's a research tool, but it's also a tool that is used, we'll say, by a pathologist. Not something you use all the time, but it is something that has a value in the clinical situation. So the lysosomes are very small. They were discovered by Dr. Christian to do, if you don't have to memorize that, let's say back in 53. And it turns out they also have a very acidic environment, and they contain lots of enzymes. So would you want the lysosomes to just spontaneously break? No, because they would chew up the very cell, that very cell. As a matter of fact, as cells get old, there's no doubt that the lysosome takes care of what we call autophagy, if you will. In other words, destroying self-destruction, autolysis. As we get older and we can't stop it, we've got less muscle fibers and etc. Lots of other things happen. But one of the things that everyone can observe, probably, maybe not if you're 20, but if you're even 30, I would say you could identify as the fact that, even if you go back to the gym, you're not going to get quite as strong, perhaps, as you were when you were, let's say, 20. What's going on here? What happened to me? Well, what happened is, I guess you could say, there's an old phrase, use it or lose it. There's some truth to that. But you might say, well, can I get it back? Well, actually, as we get older, we literally lose muscle fibers. There's other things that happen, too, but we lose muscle fibers. So even if you get testosterone, pumped up with testosterone and start pumping iron, you're not going to look like that 20-year-old or 18-year-old who doesn't even have to use testosterone, if you will, to get those muscles, because you've lost muscle. It's just the way it is. Now, I don't want to discourage you from exercising, because the more you exercise, the less you lose. So there is an advantage, but you're not going to be able to be the same. So these lysosomes then, of course, the next question is, and I don't have an answer for you, is exactly what turns the lysosome on to do this. In other words, cell death we're interested in, but nevertheless, these are the nasty structures that take care of it. They do some other things that are very important, though. When something comes into a cell, we bring it into what's called endocytosis, and it's in a little vesicle. It's wrapped in a little piece of membrane. Because we don't want to just bring things in and expose it to the whole cell. And so the lysosomes will attach themselves to those endocytotic vesicles, inject their enzymes, And what we will do is slowly break down that material. If you do a time-lapse photography of a vesicle, it just slowly shrinks and disappears, and that's through the action of the lysosomes. So they're a normal part of our cell. If they get out of kilter, though, we can have a problem. Well, one example is muscular dystrophy. We call it a muscle wasting disease, but the agent that carries out the destruction, not really you might say the ultimate cause of the disease, but the agent that carries out the destruction of that disease, the lysosomes. And that Dr. Christian Dedude was a big deal because he found the lysosomes, and he was working with people who were working with muscular dystrophy, and so it was kind of quite a big deal. So the lysosomes are very important structures. They get us in trouble. Right, so. What's next in the notes? We have to do what happened to?
We skipped right
along
earlier. Yeah, you started off out of order.
Okay, well we have to do ribosomes. That's all right. It doesn't really matter what the sequence is in some of the. Probably we should have done it shortly, but let's take a look at ribosomes. And also along with the ribosomes, I guess we're going to be talking just ever so briefly about the concept of production of protein. Earlier we said that DNA directs the production of RNA. We don't need to know a lot about it in this course. If you want to, there's plenty of information out there available. Even your book goes into things in a lot more detail than we can possibly have time for because we have so much anatomical material to do. But at any rate, the DNA directs the production of RNA. RNA carries a code, right? It literally transcribes a code in the nucleus, moves through those pores, and then finally makes its way out to the ribosomes. And actually it's an event, a special activity, because what happens is the ribosomes, even though we describe them as small spherical structures, there's actually two pieces when you look more closely. We have a large unit and a small unit. And these two units come together with a special type of RNA called messenger RNA riding right between them. Notice the word messenger RNA. So we have a message. We have a code for the sequence of the amino acids in the protein. And so we're going to make the protein. Some people can use the word in, or we can just say we're going to make the protein on the ribosome. We're going to read and write. It's called translation. But I like to think of it like reading and writing. What do I mean by writing? We're writing a sentence. Words. We're going to put the amino acids together. So we're going to read the code, and we're going to write, and we're going to make a specific protein that the body needs, that the DNA has directed its production. Maybe insulin as an example. So the ribosomes are the sites of protein synthesis, whether they are upon the ER or whether they are floating freely. This is where protein synthesis occurs. If a cell does not make any protein for export, let's just say, would it have an extensive rough ER? No, not at all. So depending on what the cell's function is, obviously a pancreatic cell has to produce proteins to take care of itself. There's always rebuilding, there's continual breaking down of the cell and building of the cell, so we're spending obviously ribosomes for that, free ribosomes for that. But we also know the pancreatic cell produces a lot of protein for export, insulin in other words, so it's going to have an awful lot of rough ER in it. All right, any questions on that? We can't see these structures with a microscope. Today when we go to the lab, if you say to me, like, where are the ribosomes or the lysosomes, no luck. We could possibly see mitochondria if we look at a special slide that's been stained for mitochondria. So you can't just look in a cell. You'd have to ask me, and I think we probably have one, not in the anatomy ones, but in the biology section of slides, we probably can find a slide that would be specific for mitochondria, and it's not very exciting because they're so small that with a light microscope you really can't see much with these tiny little specks. So it's not, as I said, not very exciting. You're going to have some fun today. Eventually, when I finish talking and lecture, you get more lecture from me. Sorry. But we're going to be looking at, through the microscope, at some cheek cells, which are your cells, and you can see the whole cell in the nucleus, and that looks pretty good. But you can't see all these organelles that we've been talking about because we need the electron microscope to really discern the structure of most of these. Your book has some very nice electron micrographs. I encourage you, if you have the book or any book that you have, to look at them. Some of the newer books, of course, the McKinley book, I'm staring at it over the, right from here, it has very nice electron micrographs. And it's interesting, if you look at the electron micrograph and then you look at the picture next to it, the artist's version, or any of these things, you'll realize, wow, how on earth did they get that maybe from that electron micrograph? So it's a lot of work, and so you can actually get a PhD not only in physiology, but you can even still get one in anatomy. And you say, how could you do one in anatomy? Because nothing changes. But that's anatomy, right? So you can do it in sort of a histology. You could get, let's say, a PhD or something by evaluating or looking at different structures and then you basically rebuild it. I don't know about now, but there's no doubt that people probably got their degrees in something by actually just literally just examining electron micrographs. Because how do you rebuild that from that one picture? In other words, if you didn't have that green picture on the right there and you were doing it, you'd say, how did you come up with that other structure? So what they have to do is what? Lots of slices. And I can tell you, I've done electron microscopy and it is slow work, frustrating too, because when it doesn't happen right after you spent hours and hours, and then you book time on a scope and everything else, being the college or university, if you're in a certain department, you pay for time, and then you go over and you look through the microscope, and if it doesn't go, man, nobody's happy. So it's not easy, but it's what you can get if you're willing to be patient. It's quite amazing. Okay, well, let's see, what else? Since I lost my place, where are we headed next? Peroxisomes and vaults and things? Okay, I don't have a picture, I don't think, of the peroxisomes, but we can just look at some of the wording. Peroxisomes are small structures, also about 0.5 to 1.2 microns. Yeah, pretty hard to really see in a microscope. I don't know if there's any preparations where you can really see them. Bottom line is they're going to be able to get rid of various harmful substances. They're going to be able to even break down fat, but they also are involved in detoxification within the liver, alcohol and other harmful materials. So they contain enzymes to convert the hydrogen peroxide, which is produced by the peroxisomes, to water. Otherwise, the hydrogen peroxide, as you can see, would destroy the cell itself. Centrosomes. Then we have to go back to our earlier picture. Centrosome consists of two centrioles. Let's see if I have any pictures. I thought what I did the other evening is, what I did is I found a couple of pictures. And so here's a centriole, electron micrograph. I didn't give you the picture. I just did this the other night. I didn't want to change the notes for this. And you can see this, by the way, is a magnification of 305,000 times. Centriole consists of 9 triplets of microtubules. You can see 1, 2, 3, 1, 2, 3, 1, 2, 3, arranged in a circular fashion. There's 9 of them, so that is kind of an incredible picture for that matter. And there's a good idea of electron microscopes and what you can actually see. And it's good, but it's still probably, you might even want more, but if you can't get too much more than that. So the centrioles, the centrosome consists of 2 centrioles that are perpendicular to each other, each one consisting of a centriole with basically 9 triplets of microtubules. What I usually say is that centrioles are basically, or centrosomes, which consist of 2 centrioles, are involved in cellular reproduction. I usually leave it right there. They also are structures from which microtubules form, and then I just leave it about that. And then we can talk also about something else, vaults. Vaults are relatively new. I don't know if your textbook bothers to talk about them. This is something we're going to hear more about. It looks like, when you look first glance, they were discovered in 1986. Let's see if we can find the slide for that. Where'd it go? I don't exactly know how they got that picture or much about it, but this is an illustration of the vaults in our cells. There are apparently quite a few vaults, as you can see. They have a size 72.5 nanometers tall by 41 nanometers wide. What do you think about that? Size-wise, even compared to the other organelles. That is small. Electron microscopes were around a long time, and it's not until 1986, at least it seems, that people accept the existence of vaults. And so some books don't bother talking about them very much because we don't know an awful lot about them. But there is a probable function. they probably move RNA from the nucleus to other parts of the cell. Remember, we found out the DNA directed the production of RNA, and I said it moved out of the nuclear pore. So they're guessing right now, but they think that the vault literally docks, so to speak, with the nuclear pore, and then moves the RNA out, let's say, to the ribosomes or to some other location. There's no real, so we're guessing now, because again, they've discovered them. Well, you can imagine, based on their size, they are very hard to study. And so there's conjecture, you might say, right now. Medically, there's a great interest in them, because wouldn't it be nice if we could actually take the vaults and I'll get them to move drugs, if you will, to specific locations within the cell. Be a great delivery system for drugs, and as you can say, even for DNA. So we could